listening to Sasquatch Syndicate.
911, what are you reporting?
We got someone or something crawling around out here.
Did you see what it was? Was it a person or an animal or?
I can't tell. All I know is that my central light came on and I just happened to glimpse and see this thing running across the yard.
And all the ape believers don't want any of the paranormal believers to say anything because they're all whacked and screwed up and we don't want them. And all the paranormal believers don't want, go to the ape believers saying, well, you're all closed-minded. You're not open to the fact that it does this and it does that.
And I look over my left shoulder and this creature is running through the woods and it's bulldozing the brush down. And I knew, man, this thing is going to get me.
Welcome to Sasquatch Syndicate. I'm your host, Chuck, out in Seattle, Washington, along with Paul in Portland, Oregon. Thanks to everyone for listening and those following us at SasquatchSyndicate.com and on our social media outlets. For those participating in the Sasquatch Syndicate monthly t-shirt giveaway, this month's winner was Aaron McCabe from Snowflake, Arizona. So Aaron, thanks for listening to the show, and please check your Facebook for winner notification. Just a quick announcement, Sasquatch Syndicate will appear at Bigfoot University April 21st through 23rd. That will be held at the Baymont Inn and Suites in Bremerton, Washington, and you can pick up your tickets at teamsquatchingusa.com. Happy April, everyone. We hope you're able to get outside and enjoy the spring season. We've got a great show lined up for you tonight. With us on the podcast is Dr. Todd Disotell. Dr. Disotell is a molecular primatologist from New York University and is well known for validation of DNA studies worldwide. So Dr. Disotell, welcome to Sasquatch Syndicate. And if you wouldn't mind sharing your background and your current position at New York University for our listeners.
I consider myself to be, the title I usually give myself is molecular primatologist because I basically study anything related to DNA in any of the primates, not just in humans. Most people think anthropologists, just study humans, but in fact, I'd say 80% of my work is in non-human primates. And so we use DNA to infer evolutionary history, population history, the history of natural selection on the genomes of different groups. We also just use it as a tool for identification and finding relatedness. Say, for instance, who's the father in a group of wild primates, or how closely related are all the individuals within a group. So basically, anything that DNA can inform us about the biology and history of any group of organisms.
So as you began your academic career, where did you really start in the field?
I was an undergraduate at Cornell University, where I basically triple majored in math, computer science, and anthropology. And I went to Harvard for my PhD in biological anthropology. I started out studying anatomy and paleontology and even did paleontological expeditions to Pakistan and switched about halfway through into molecular genetics so that I could use DNA to tease apart evolutionary history rather than the fossil record. And now, of course, we can kind of do both of those simultaneously with some of the new technologies.
Have you always been at NYU, and what's your current position?
NYU was my first job straight out of graduate school. In fact, I was offered the job before I had defended my PhD thesis, though between starting it, I did finish my thesis and moved to New York in the fall of 1992, and I've been at NYU continuously ever since.
So Dr. Disotell, I know a lot of our listeners are probably not aware of what you wrote your thesis about, and it might give them some perspective on why you've kind of become the chosen primatologist on a lot of different TV documentaries and shows. Could you share a little bit about that?
So I went off to Harvard. I was very interested in fossil apes, including Gigantopithecus, Shivapithecus, Gigantopithecus, and all of the Miocene fossil apes, which basically existed from about 5 million years ago to about 23 million years ago. And one of the world's foremost specialists on those was at Harvard University. And so I went there to study with him, and that's how I ended up in the field in Pakistan looking for fossil apes. And I actually found several fossils while doing that. But I wanted to use the fossils to reconstruct their evolutionary history. And at the time, it was kind of muddled. I mean, many people still thought chimpanzees and gorillas were more closely related to each other than chimps are to humans, which we now know to be the case. And so my advisor suggested I use the morphology of old world monkeys to figure out what features of the skeleton allowed us to reconstruct an accurate evolutionary history of old world monkeys, which are the next closest things on the evolutionary tree to apes. And I found out I was having trouble doing that because the way people interpreted morphology and fossils often was arbitrary. You could have people studying exactly the same groups coming up with completely different estimates of their evolutionary history based on how they interpreted both the fossils and the morphology. And it was at about that time that new DNA technologies came into being, particularly something called the polymerase chain reaction, or PCR, which kind of, for want of a better word, it democratized molecular genetic research. It allowed people without deep training and really expensive laboratories to actually begin to get and sequence DNA. And it made it quite easy. So I was actually just sent over to a DNA lab. My advisor had met the person who ran that lab at a cocktail party, and they found out just over drinks that they both were reconstructing evolutionary history, one with DNA, one with fossils. And so he sent me over, and without any background in biochemistry or organic chemistry or any of that stuff, I slowly, actually quite rapidly learned how to sequence DNA. And so I sequenced the DNA in a variety of old world monkeys, and the evolutionary tree that I inferred from that was actually very significantly different from that all of the morphologists had ever put together. And in fact, it led to a new grouping of these monkeys. We had to rename some of the genera that these monkeys belonged to, because it turned out that monkeys that looked extremely different from each other were in fact very closely related, and another group that are almost indistinguishable from each other were extremely distantly related. Because again, morphology is very difficult to interpret. Where DNA, it's kind of simple. You have A, C, G, T. You're not making a judgment, oh, that's medium and that's small. And we know the features. We know DNA is passed directly from generation to generation. Whereas a morphological feature, we don't really know that that same cusp on a tooth in two different organisms is actually the same thing deep down. You can have similar things evolve in parallel or convergently, and that very frequently misleads us when we are using morphology to interpret or try to infer evolutionary history. I mean, think of, look at a shark and a dolphin and some of the other fish. They all look extremely similar to each other, even though, you know, one is a cartilaginous fish, one's a bony fish, and one's a mammal, yet they look remarkably similar to each other.
So is that kind of like the Skywalker gibbon they discovered over in China? I think it, if I recall, it was very similar, and it looked exactly like a different type of monkey, but when they did the DNA analysis, it was actually a completely different species.
Yeah, and so we can also use DNA. One of my former PhD students, who's now a professor at the University of Texas, San Antonio, he studied a group of nocturnal primates, and you can barely tell these guys apart. I mean, they look so similar to each other, yet they turned out to be different species and even different genera. So how similar something looks actually is a very poor indicator of how closely related it is to something else.
So Dr. Disotell, I guess in this subculture, you've really become kind of the go-to person as it relates to DNA. I was just curious, why do you think you've become that person?
Well, partly because I'm one of the only people who's actually willing to do it.
And I guess I just want to take a moment and thank you and just kind of distinguish that, because you are one of the few scientists that do step up and are willing to do this. And as a result, I think you've also discovered new species and probably inspired a few others to do the same.
My colleague Brian Sykes at Oxford University just did, a study, 2 years ago now or a year ago now. But very few actual, bona fide scientists are willing to get near this stuff. Ketchum pretended to, but, she's not a trained geneticist, and her results are rather unorthodox, let's say, as are her techniques. But I agreed to do it years ago for the National Geographic Channel. They had an interesting story, and it turns out, and I always tell people this, that, I study DNA to try to discover new primates or figure out what primates we have every day. That is the kind of research we do in my lab. So having an unknown sample and figuring out what it is, that's just absolutely day-to-day activity for us. Usually, is this a new subspecies of baboon, or is this a new species of gibbon, or something like that? But the techniques that one would identify Bigfoot or Yeti or whatever with are exactly the same, this very same scientific techniques that we do. So I've been willing to do it. I've probably done, I've looked at hundreds and hundreds of samples, but I've always done it in the context of some kind of TV show, because to me it's very important to explain what the science is and how it works. Because if you look at 90% of all those cryptozoological TV shows, they're ridiculous. To me, they're basically unwatchable. Oh, did you hear that? Or, oh, look, there's a broken sapling. That's their evidence.
So as someone in the field that really does understand DNA collection and analysis, as you look at some of these TV shows, and I was picking on Monster Quest the other day, where they had a blood sample or a hair and they were breathing and touching it. How does that strike you?
So I like to also, explain how the scientific method works, and that you need to bring a level of skepticism. Every whoop you hear in the forest is not necessarily something calling to you. Every broken tree, gravity has an effect on things out there in the forest. And so, you have to, I try to say, well, what's the most likely explanation for this? That scat sample there, bears do shit in the woods all the time. All sorts of animals do. And so if you find a scat sample, or if you find a hair stuck to the side of a tree, it's most likely, and every single time I've ever done it, it's something that's explainable. The same thing with Brian Sykes' study. He took something like 30 samples from all over the world, and every single one of them was explainable as a horse or a wild boar or a human or a bear or this or that. And, he collected samples from people all over the world, and every one of them turned out to be something that lived in that region.
So if you wouldn't mind, let's just go back to the stuff that we do know existed. Gigantopithecus, you'd done a thesis on this, and I know a lot of people understood that teeth were found in apothecaries back in China.
Teeth and jaws. So there's thousands of teeth, but only a handful of jaws. And so, I mean, it was named Gigantopithecus a long time ago. But a long time ago, probably 2/3 of the primate fossils that were named a long time ago have been renamed after being more properly discovered. So it had big jaws and big teeth. We have no idea how big it is. There are some things that have bigger jaws and bigger teeth. I mean, if you look at some of the robust paranthropines, what used to be called robust Australopithecines, they have enormous jaws and enormous teeth. Some of their teeth are actually very similar in size to those of Gigantopithecus, but they were 3.5, 4 feet tall. You know, they weren't giants. You know, maybe the largest of them might have been 5 feet tall, if that, but probably under 5 feet tall, with enormous jaws and teeth because they were eating a very hard, tough diet.
So, Todd, back in February and March, we had both Dr. Bindernagel and Dr. Meldrum on to talk about, the Gigantopithecus hypothesis. And just curious, coming from your background, I was just curious, I was just wondering, kind of what you think of the Gigantopithecus theory, the hypothesis, and whether or not it fits.
To say that Gigantopithecus was some kind of 8-foot monster, we have zero evidence from that, and we cannot easily extrapolate just from jaw and tooth size to that.
So, Dr. Disotell, what could DNA really tell us about a species? Can it tell us the diet? Can it tell us about the climate it was in, what foods it likes to eat? What can you really discover from DNA?
Well, so there's different things we can do with a DNA sample. Some of it is just quick, mere identification. And we can, try to identify it very precisely or just very broadly. So sometimes when I'm doing these samples, I will use the mitochondrial genome because mitochondria DNA, which is only inherited through the maternal line, there are 100 to 1,000 copies of the mitochondrial DNA in every cell and only 1 or 2 copies of the DNA found inside the nucleus on the chromosomes. And so for just a hair sample or a forensic sample or just a tiny blood spot or something like that, we're far more likely to be able to get the mitochondrial DNA out of it, given that there's 1,000 times as much mitochondrial DNA in the sample as the other stuff. And with that, we can then get pretty close. And sometimes I'll just do sort of a quick analysis, and it'll tell me it's ursid, it's a bear, but I won't go even further to see like which species or subspecies it is because it's a bear, it's not Bigfoot. So you can do a quick analysis like that to say it's, some kind of cervid, it's an elk or a deer or something like that. And so that kind of analysis is very quick and easy to do. And that's basically just genotyping a portion of the mitochondrial genome, which different groups of animals have different sized parts of the mitochondrial DNA. So some of them might be 350 bases long, some might be 450. Humans are 453 at this particular place that we look at. Some of them are up at 600 or 700, so we can do that without actually even having to sequence it down to the individual bases. If we want to know precisely what species it is, we need to sequence the DNA, and then we compare it to either our database, or if it's not a perfect match in the database, we can then basically infer an evolutionary tree from it and see what it's most closely related to.
So in terms of the sequences and the DNA analysis of species, how many sequences do you have in the database today?
I just looked it up a half hour ago. Has 43,732 mammal sequences in this database. Most of them are human, but there's about 9,000 non-human mammal sequences in this database. So basically, every genus and almost every species we already have in the database. So if I sequence a portion of the mitochondrial DNA, I can tell you exactly what it is, or I infer an evolutionary tree and I say it's some kind of macaque that's not yet in the database, but, it's a macaque, or it's a baboon, or it's a gibbon, something like that. So that's if we're just looking at the mitochondrial DNA.
So I know, DNA testing is getting, certainly cheaper, but if you had to do the whole genome, what's that process like?
If we want to spend a lot of money, we can actually sequence, and we have a good enough sample, we can sequence the entire genome, and from that we can sometimes pick up some kinds of clues as to what its diet might have been like, how cold adapted it is, what the hair color and eye color are, some of those things we could actually get out. And as we learn more about the connections of the genome to the body, the genotype to the phenotype, we'll be able to define those even better and better in the future.
So as we think about DNA evidence and gathering evidence, what would give you the most information about the creature? Would it be scat? Would it be hair? Would it be saliva? What type of tissue or what type of sample would give you the most information?
If you get a scat sample, I can actually tell you precisely what its diet was in the last few days, because we can sequence all of the DNA in that scat sample, and that will be every bacteria, every plant, every animal, plus the thing that actually left it behind. And so I can tell you it was eating these berries, these plants, it had a couple frogs, it ate a few termites, and it ate this. So we can actually use DNA analyses to try to infer what they ate, at least short enough ago that it's in, their scat sample.
So in terms of preserving collection samples, would your preference be hair? Would it be tissue, saliva, scat?
Fine if it's fresh, but then it also has to be preserved well, and so hair is actually probably the safest and the easiest.
So if you wouldn't mind, maybe we can just talk about the steps to data collection and, of course, DNA collection. If you wouldn't mind just kind of sharing what you might want to have in your kit and maybe the best steps on collecting evidence.
So the very first thing one should have, the tool they should have, is a camera. I want to see, okay, they say they found a hair, on the ground next to a footprint. I want to see pictures of that before they've even touched it. And, getting good pictures, and then if you're getting a good picture, you also should record where you are at, so, GPS coordinates, or mark it well on a map. When you take pictures of anything, you should try to put some kind of ruler or marker, or even just a coin or something down on the ground for scale, so that we can judge how big something is in the picture. And that's before you even get close to touching it. Now, it's best, especially if you're collecting hair, it would be great if you had on a mask, because just breathing, you're breathing out tiny particles, and those particles contain DNA. If you've ever seen any of the famous, high-speed pictures of people sneezing and stuff, the horrible spray shooting out, 5 or 6 feet in front of them, all of that's got potential DNA in it. You should definitely try to be wearing new, fresh, gloves, latex or something if you're allergic to latex, one of the other materials. You should collect it with some kind of sterile tweezers, and you can have a tweezers with you that you basically just put under a lighter for just a few seconds, and that will sterilize the tips of the tweezers when you pick it up. And then you should put it into some kind of sterile container. A tube, or one of the best things for this is actually glassine envelopes. It's kind of like a wax paper or envelope that stamp collectors and other people use, to get the hair away from the environment and sealed off from the environment so that it's not going to collect DNA from something else. Because if the hair is fresh, it's likely it's a very good source for DNA. If it's older, that DNA is going to be degraded, and any kind of contaminating DNA that you, the collector or people looking at it, their DNA is going to be newer and higher quality. So when we try to get DNA out of it, we're likely to get the high quality contaminating DNA rather than the DNA, the endogenous DNA that's from within the hair sample itself. If you're collecting scat, again, you want to make sure you collect it with something sterile. One of the best things is get a bunch of sterile tongue depressors that are, they're sterile inside an envelope. Right before you collect the sample, you tear it open. It's sterile. You collect the sample into a tube that you can seal. You can put it into basically pure ethanol or, say, rubbing alcohol. Isn't bad. There is a substance that we use in our research called RNA later. It's actually quite expensive. It's several dollars an ounce, but it really preserves DNA very, very, very well. And so when we go off to Africa and South America and Asia, all my people are carrying, dozens and dozens of tubes that seal really tightly, because if you're using anything and you put it in a tube, you don't want the tube to leak. So we use special tubes that have a little rubber O-ring in them. So when you screw on the lid, it screws down tightly, and there's like a rubber gasket in it, and so they just don't leak as easily. The other thing you need to do with both any kind of samples is you need a Sharpie. You need to write down as much information of it as possible, especially if you're collecting several samples. A hair looks like a hair looks like a hair. If you've collected 5 hair samples in 5 days.
So we talked earlier about, potentially wearing a mask before you collect so you're not breathing on it. But would things like hairnets or a hat help?
That might help. In the ideal world, you would have on, a full, Tyvek suit. If you've ever watched any of the police procedurals, those guys going in, they're in full Tyvek suits because your bare skin gives off skin cells. You're losing hairs constantly. As you breathe, you are exhaling DNA. So the cleaner and tighter you are, the better. And, again, don't, get down on your hands and knees right on top of the sample and breathe on it as you're, even if you have a mask, try to stay as far away from it as possible as you collect it.
So, Todd, let's just hypothesize, but let's say you have a DNA sample. How are you going to be able to tell that this is a Sasquatch? Would it take on a primate with peculiar characteristics? Like when you look at it, in terms of DNA, and especially, something that's undocumented, how are you going to be able to classify really what that creature is?
Well, so this is, it's an interesting question, and it's every now and then in my lab, we'll sit around with a few beers and, discussing, what would it be? And, many people have made estimates, so there's the whole Gigantopithecus crowd. There's the whole relic hominin, like a Denisovan or a Neanderthal or, some other unknown ape crowd. But all of those, so one, it would fall outside the range of any known species. So, so one of the things, say, on $10 million Bigfoot bounty is, that was part of our definition for the DNA evidence, is that it had to fall outside the range of any known species. Because if somebody had a pet chimp and it just got too big and unruly for them and they just sent it off into the woods, you would find neat footprints and you would hear and see weird things and find hair and scat, and it would turn out to be ape, and but it would be chimp. So it has, presumably I always work on the premise that it's a primate if it's there. And again, I can talk about what I really think in a few minutes, but it would presumably be primate, and even if it's not, again, if it's not in the database, we would then infer an evolutionary tree. Where does it fall? So the Gigantopithecus people who think it's a relic Gigantopithecus most likely belongs to the lineage that gave rise to orangutans. Now, humans and orangs split about 14 million years ago, and so we would, if there was a sample that fell on the orangutan line, but it wasn't an orangutan, and it split from orangutans several million years ago, that would be a really strong contender for a living unknown ape related to the orangutan lineage, which most people would be at least willing to consider, might be a relic Gigantopithecus. Because we do have Gigantopithecus fossils in the last several 100,000 years. So, the most recent fossils are somewhere between 200,000 and 300,000 years old of Gigantopithecus.
Todd, does it ever, just drive you crazy? Certainly, here you are, you wrote your thesis on the subject, and, besides, jaw and teeth, when they were mining this for the apothecaries, do you think that they ever found anything else and just didn't know what they had or misidentified it?
We got a few spectacular Lucies and some of these other things, but the vast bulk of human fossils are jaws and teeth, just because they're so dense and they last. Those bones last really, really well. And they're far more recognizable. So, yeah, people puttering around in caves in India and China might have found more things, but they were looking for teeth to, grind up into powders and stuff like that. And so they probably, if there were other things, they would have just left them behind.
So, Todd, if you had to go back on the hunt for Gigantopithecus, would you go back to, where they found the jaw and the teeth, or, or do you think that there's, still bones and skeletons that still exist? Like, what's your thoughts?
Well, people are looking for them. There are paleontologists working in, China and Vietnam and India and all of those areas, and, they just haven't come across them yet. Usually, they do find, fossil humans and fossil gibbons and fossil orangs and stuff like that, but there's been very few. And, few people, it's those areas have been hard to do research in for the last 50 years. China and Vietnam are just sort of opening up to Western researchers collaborating and working with them more often than in the past. So it's more likely sometime in the near future that something might be discovered, just because now there are more and more people doing it. And there are, with the Chinese economy improving and building universities and training people, there's going to be more people looking for that stuff. So, yeah, I think it could pop up at some point in the future. Where I did my field work was in areas of Pakistan no sane Western person would go near today. So I doubt there's much paleontological work being done there. But there are areas of India and China, and again, that whole Southeast Asian region that could turn some of this stuff up. Where it's most likely to turn up is probably in a museum drawer. People misidentifying it, 75 years ago. And if you've ever been to a research museum or a natural history museum, what you see in the glass cases in the hallways is about 1% of what they have. They have drawers full of thousands and thousands of bones and teeth and jaws and skulls and skins, in the back rooms where the research is carried out. And tons of important fossils have been found there because they were misidentified. Somebody was out looking to study bears, and so they collected all sorts of bear fossils in caves, but they accidentally also found some human fossils. But since they were bear experts, they didn't really recognize what they were. And so they're just in the junk drawer, if you will, from that expedition.
So if you were to give any advice to those with time on their hands, like a graduate student or a researcher that maybe is, working in the back room of one of these museums, what would you say?
Well, usually is what happens is a researcher or a graduate student or somebody is carrying out a project on something else, and so they go to look at the collections, and that's when they figure things out that were misidentified. One of the most famous recent cases of that is, the famous Lucy skeleton of Australopithecus forensis. It was discovered in the 70s, and it's been on display, and there's casts of it all over the world. Almost every museum you have that has a human evolution exhibit has it there. It turns out one of its vertebrae was misidentified when they first discovered it, and it belonged to a baboon, because there were also fossil baboons at that same site. And baboons and humans, our skeletons are extremely similar. And vertebra are very complex, and not a lot of people are experts at them. One of the world's best vertebra experts happens to be a colleague in my department, and he recently figured out that one of Lucy's vertebra was a baboon. So I don't think curators are going to go look for Gigantopithecus for you. They're busy just running the show, but there's plenty of other researchers who might come across it.
So Dr. Disotell, you'd mentioned the evolutionary tree, and for our Sasquatch Syndicate listeners, can you expand on that? Can the evolutionary tree kind of tell us, can the evolutionary tree depict, whether the species interbred with other species, if there were any genetic mutations along the way? Can you expand on that a little bit?
Well, so if we're going to place a DNA sequence or a series of DNA sequences onto an evolutionary tree, it immediately tells us two things. It can tell us what something is most closely related to. So, for instance, somebody just brings in a bone fragment from the forest, and somebody sequences it, and they say, this is a cervid. It's some kind of deer or elk. We're not going to go further in trying to figure out if it's Bigfoot or not, because it falls on the wrong. It doesn't even fall within the primates. It's not a primate. And so it can tell us what is most closely related to the sample and how long ago that relationship is. So basically when the branch leading to the sample and the branch leading to all of the other branches of the tree, when did they split? So did these things split 100,000 years ago, 14 million years ago? When did they split? And that would also help answer the question of sort of what could it be. If it's a human-like sequence that split a couple 100,000 years ago, well, that could be a Neanderthal or Denisovan. And now that we know the Neanderthal and Denisovan genomes, we can actually then compare that sequence. How closely related? Is this just a particular type of Neanderthal or Denisovan, or is it something significantly different? Is it a new species of early human-like hominin?
So Dr. Disotell, obviously, commercial DNA testing is very expensive, although public testing is, coming down, services like 23andMe and Ancestry.com. And, while they're widely available, unfortunately, they're not the correct avenue for wildlife studies. But if there are listeners out there with samples they wish that they could have, DNA tested, what would be your recommendation, and who would you have them contact?
There is a commercial lab up in Canada that does basically, so this kind of analysis is called forensic wildlife analysis. So people often, so somebody is studying wolves or coyotes and they find a bunch of samples. They want to verify, are these in fact from wolves or coyotes? And if they don't have a lab or a colleague, they can send it to one of these forensic labs. Sometimes archaeologists will send samples to them because they're not quite sure what the bones or the hair that they found are. If I'm not mistaken, they charge $300, $350 a sample to do that kind of analysis, because there is overhead involved and stuff like that. And they're set up to do that. They are a commercial lab, kind of like 23andMe, who does this every day. While I can do it, I don't look for samples. I'm not out there trying to. So one thing I want to sort of make clear is while I have done tons of these samples and I have, the reputation as being the Bigfoot DNA guy, I'm not out there looking for it. I don't consider myself a Bigfooter in any way, shape, or form. I'm a molecular primatologist who can test samples, and under the right circumstances, I have done that for different networks and producers and people, but I am not a commercial lab out there looking to have this done, to have people send me samples. I get dozens of cold calls and emails a year. I found the sample. How do I send it to you? I'm like, I'm not interested in doing that, but here's the name of a couple companies that will. And, all these people think, oh, I found hair on my weekend Bigfooting expedition. Now I'm going to get it analyzed. Well, the cost is something that makes people then think twice about it. If somebody was independently wealthy and they wanted to test every hair they found in the woods, there are companies that will do that for them. And then, again, there's different layers and levels of analyses. Just to ID it is one thing. To do more of the genome to figure out, is this a hybrid with something else? That ramps up the cost of being able to do these kinds of analyses, and those also require higher and higher quality samples. A single, 6-month-old hair that's been exposed to the elements, you're probably not going to get a whole genome out of. And whole genome analyses cost thousands of dollars to do.
Okay, so Todd, let's just kind of get back to it. Somehow, and maybe I'll just do the backstory. So I'm at a conference, I'm standing there, and two guys walk by and they're talking about DNA. And the one guy says, well, I'm not going to believe it unless Disotel says it's true. So just kind of going back to, here you are, you're the judge. What was it that really kind of propelled you into that kind of role for DNA analysis, for this subject? And was it your thesis? Why do you think that you've kind of assumed that role in the community?
Being in New York City, where, the television Nat Geo was, and they cold called around to a few, I think they called Columbia and they called NYU, and I actually spoke to them, and, so we had at that time just published a series of papers basically announcing the discovery of a new subspecies of chimpanzee, and we did that work from hair. And about 2-3 years later, we did the same thing with a new subspecies of gorilla, which we did from feces or from scat samples. Those studies were actually done in my lab. So my lab was known as a lab that could identify new species and subspecies. And, as I said, the actual scientific technique, what I do in the lab is identical for any sample. It's DNA after all. So once I extract the DNA out of the sample, I usually just label the tubes, 1 to 8 or A to, H or whatever, and we work them through, and then in the end, we're like, okay, what was sample A? Where did that come from? It's, that's American black bear. So again, I agreed to do it for them, partly because I wanted to really try. I had seen so much bad stuff, for blurry pictures, crappy footprints, ridiculous theories, millions of eyewitness accounts. They're next to worthless without actual data. And so I said, yeah, I can do that, and but I want to be able to talk about what the science behind this is, and they let me do that. And to me, that's one of the things I do as an educator. Is I try to not just educate my students, but the general public about science. And believe me, we definitely need that, especially in this day and age.
So Dr. Disotell, of course, a lot of the evidence that, we hear about Sasquatch and Bigfoot is anecdotal at best. And certainly, in terms of eyewitness encounters and footprint evidence and, those types of things, what would be maybe some of the more prevalent things that you could test? And why do you think that the scientific community doesn't necessarily, enter the field or embrace this?
Well, we have, from the scientific method point of view, anecdotal evidence is that there's nothing there. There's nothing we can test. So it's, it's more or less inadmissible as scientific evidence. People's stories change, mass hysteria. Look, they burnt young women at the stake in Massachusetts in the 17th century because everybody saw that they were witches. And there's tons of, hundreds and hundreds of sightings of things, usually sort of following after, a famous case. All of a sudden everybody starts seeing, things. The number of Bigfoot sightings after Patterson-Gimlin shot up enormously. The Loch Ness Monster sighting shot up enormously after King Kong came out in the 30s. But there's not one thing there that a scientist can study. It's like everybody says this, it's 8 feet tall and it has glowing red eyes. What mammal has glowing red eyes? But these stories just keep going. So those are more or less meaningless. Footprints potentially could be meaningful. They're pretty hard to do well and to study properly. There's a lot of things that leave what look like giant human footprints, bare double strikes and all of that stuff. All of the stuff about the mid-tarsal break, scientifically. So, footprints are also a very poor, if you will, source of data.
Yeah. If you think about the, so I guess, we've reached this peak of humanity where I think like the outside is treated as the place that you leave from your house to your car. There's less people outside. They're more connected to their phones and devices. And so I think we've kind of peaked on the number of people outside. So you couple that with something like maybe the giant panda, which back in the day it took, 60 years to discover. Do you think that there's still, we discover, undiscovered primates quite often, not as maybe often as you would think, but like what are your theories that if it, is it possible? Could it still be out there in maybe the Northwest or, in these?
So I have never said it does not exist. What I've said is no evidence of its existence yet exists. To me, zero scientific evidence has been presented that is credible evidence for its existence, and I'm willing to say that. I will never say it can't exist. I think it's very unlikely. One, there are literally thousands of people looking for it. There are camera traps. Everybody's got a camera or a phone on them now. We still don't have one single credible photograph or video of it yet. That, to me astounding, given the number of people looking for it. The amount of evidence these things do, defecate and urinate and all of these things. There should be samples out there. Nobody has produced one yet to date. And then there's the concept of basically, population viability analyses, which is a real biological field. It can't be one or two. It's got to be hundreds of these things, or they would be extinct. A population of just 10 or 20 individuals is going to go extinct by accident with an extremely high probability. That tiny of a population with a drought or a really bad storm or a really bad fire, you could lose that entire population if it's small. So there has to be many of these things if they existed. And why hasn't one single bit or bytes worth of data been collected to date? If if it's out there, I think they should have found it by now. And there are some very good people who are looking carefully, and they haven't yet found it. There are a lot of super amateurs who could potentially find it but haven't yet found it. But part of the other problem is there's just so many hoaxers and hucksters out there as well, some intentional, some unintentional. And given that there's literally now an industry, if you will, and a profit motive, that makes it a little more worrisome. People will say things for money. People will do things for money, or just to trick people and fool with people and stuff like that. If back when I was a teenager, if I had heard that a Bigfoot hunting team was coming to go camping in the woods near me, I guarantee you we would have been out there every night and doing things and making fake footprints and stuff like that. So there's just to me it's incredible that if it's out there, that they haven't found it yet.
Yeah, Todd, we're really excited that you're doing this. Your level of skepticism that you bring to this is really refreshing. And as you know, if you're trying to bend the facts to, prove a theory, of course, we know that that's not science. But Todd, why do you think that the other scientists shy away from this topic? It would seem as a scientist, at least even for the sake of discovery, that they'd want to do these tests. So in your estimation, what is it about the topic that scientists shy away from?
The bulk of them are pretty conservative. They're very worried about their jobs and their reputations amongst their colleagues. I think I had established myself as a scientist before I did any of this stuff, and so I don't think it's hurt me in the broad scale of things, but it could, and there are people who do think it's a waste of time and frivolous and all of that stuff. But I make the argument, and I've actually gone and given lectures at universities about, science education and science outreach, and this is one way of doing that. So I've done the back of the envelope or back of the bar napkin calculation that as a professor, I will lecture to 10 to 25,000 people in my lifetime. Students, colleagues at conferences, maybe a public speech lecture at the American Museum of Natural History or something like that. But the tens of thousands of people will hear me. One episode of $10 Million Bigfoot Bounty had 900,000 people watch it. So if you want to get at a broad audience and a public audience, this is unfortunately at the moment the best way to do it. You don't have hundreds of thousands of people follow you on tour when you go and give lectures at museums. You have hundreds of people show up, maybe.
Yeah, and I find it fascinating, and maybe it's just, humanity. But, if you think about people staying away or shying away from somebody, due to their belief system. I would think, and by the way, I'm just going to tell you this, I like the Mohawk, but I would think that just as many people may shy away or stay away from you in the scientific community just because you wore a Mohawk, not necessarily because you were testing Sasquatch DNA evidence. So I guess it's just, an interesting observation in humanity on, why people shy away from a subject, based on a stereotype belief system.
It's fine if they shy away, as long as I can continue to publish real scientific papers and get grants and do research. I don't care what people, think. I mean, one of the reasons I think I'm in academia is I can wear shorts 8 months a year and T-shirts and tank tops and sweatshirts and jeans, and I don't have to, wear a uniform or a suit or something like that. And the Mohawk was just something I did for a while. I actually had gotten rid of it, but the executives at Spike wanted it back. So if I hadn't had it for about 2 years when they recruited me for that show, but they requested I grow it back. And the night it aired, which meant Dean and I didn't have to do any more interviews or publicity tour stuff, the night it aired, I shaved it off. And I actually flew off to England the next day for a semester sabbatical at Cambridge University.
Yeah, I thought maybe you started that down in Pakistan and, you had that persona of the American badass or something like that.
No, there we wanted to blend. There you didn't want to really stand out. We had to try to de-emphasize, even though it's not like it is today, we still had to de-emphasize who we were. So no, I grew it while I was a professor at NYU, and it didn't affect anything there.
Yeah, and we were talking to Dr. Meldrum just how, people really come out of their shell after they get tenure. You really understand who those people are. But just curious about your observations, with those being conservative, before they get tenure with universities.
Well, it is a little freeing, knowing that you can, push boundaries, explore boundaries that you might not have done while you were deathly worried about every single person in your department, in your university, in your field's opinions about you. You're going to play it far safer than most people would. But, I did my first one pre-tenure, and I actually had the Mohawk while I was being evaluated going up for full professor, which I kind of took it. To me, it was kind of like, well, if they give me full professorship, even with a Mohawk, it means I truly earned it. So it's, kind of like the peacock's tail. I
Right.
purposely had this display or this ornament that actually, evolutionarily speaking, is not very good because, the leopard's going to come after you more easily. But so it was kind of, and it was just fun to have.
So Todd, of course, this episode follows part 2, Building a Citizen Scientist. And we had spoke with Dr. Meldrum about his cast collection there at Idaho State University. And, he is the curator of hundreds, if not, thousands of casts and Bigfoot evidence. And one of the things Dr. Meldrum shared is that they're planning on doing some digitization of the catalog, for those prints. But when you think about, just archiving, physical evidence to the digital catalog, do you have any concerns? And, what do you think of the digitization age?
Yeah, I'm a big fan of it because it does help democratize it. And, I don't mind amateurs trying to do research. I think it's a great thing if they try to do research. And, if amateurs can actually download, the bones of a chimpanzee and an orangutan and print them out on a 3D printer, and so they can study them when they go out in the woods, they're not going to think, a deer limb bone is Bigfoot. They're going to be able to learn that it's a deer. I mean, one of the things my friend Stacy Brown, who was on the show, one Christmas, I actually sent him a 3-foot-tall replica human skeleton just so he could start to actually learn some anatomy. Because most of these people, they don't know any anatomy. I mean, a good hunter out in the forest, they find something and they'll say, oh, that's elk, oh, that's coyote, oh, that's bear. They can do it. And I think anybody who wants to call themselves a Bigfooter, and I don't call myself one, but anybody who wants to call themselves, they better have some anatomy books on their shelf and some cards, so that they can look and compare. It's not that difficult to identify the difference between bear bones and primate bones. In fact, there are several things out there to show you precisely how to not call the police when you find a bear paw out in the forest and waste their time.
Todd, you had spoke a little bit earlier about grants, and for the citizen scientists out there or the average Joe, looking to expand their research, how hard or easy do you think it would be for a non-scientific entity to obtain a grant? And are there any grants out there that you think, the average citizen could go after?
Well, there's really no easy grants to get, and most of the scientific ones, I think a citizen scientist would have great difficulty doing, because, they're hyper-competitive amongst the scientists, and so the real big scientific grants, they actually look at they want to know, where did you get educated, how many papers have you published, how good is your research, what did you do with the last grant you got. They're going to know that. So, I think the only place people might be able to get that kind of stuff is they need somebody and there are occasionally these guys pop up who are willing to, donate and say, I will fund a $30,000 expedition to here if you guys go looking for Bigfoot. But you're definitely not going to get one from the National Science Foundation or NIH or anything like that.
So Todd, speaking of, the DNA archival, and since we have you here, just thought I'd ask, there are a few new programs like Russia's, new ARC project. And I believe that, Britain's even doing the same thing where they're archiving not only animal DNA, but, seeds and plants and other, vegetation as well. But, what do you think of the global interest of, this DNA archive, and is NYU participating in that?
That's done. Basically, the United States government actually runs, through something called GenBank. It's one of the largest it is the largest genetic database in the world. Not every single country fully participates in it, but anybody from anywhere can access it. And basically, any paper or study now is basically required to put their data into it. So if I'm going to work on a bunch of baboons, I have to put every base that I sequence that I'm going to, if I want to publish it, that all has to go into this database so that other scientists could cross-check it or use it or build from it. And so that already exists. And there's several other databases like this, but that's by far the biggest one. And most of these other projects, the Tree of Life project and those kind of things, all of those data are also in GenBank. It's just they catalog it differently, or make it more easily accessible. But those data are collected and out there. So all sequence data being collected nowadays basically goes into these databases, unless it's, secret drug company research where they don't want somebody to know, what they found in Finland or in Iceland or something like that.
Yeah, I find it interesting as you were kind of talking about that, just the past barriers of research in China as a Westerner and things like that. I was just wondering if you had the same geopolitical boundaries as scientists as, we hear about.
Most of those have disappeared and evaporated. NYU actually has an NYU campus in China. And so we have Chinese students coming to New York City, and we're sending students from NYU to China now. And, at least on the internet, that's not behind, the DNA databases are not behind the Great Firewall. So Chinese scientists can search these databases. They can add data to these databases freely. And so there's basically no boundaries.
So just getting back to DNA, I guess, with your years of experience, what do you think the problem is right now with just DNA around Sasquatch? And
the problem is samples. There are no samples yet. So, that's the start and the end of the conversation, really. Without samples, we don't have Bigfoot DNA yet. So we can't really discuss it.
So there's been a few, what we would, I guess, as citizen scientists think are credible, DNA studies about this creature. And of course, I've heard a couple times you kind of discredit that, but can you kind of explain why?
So they did something called next generation sequencing. So that's basically where you sequence all of the DNA in a sample. But unfortunately, when you sequence all of the DNA in a sample, you get all, every bacteria, everything that lived in the soil, if it was a soil sample or if it was a sample sitting on the soil, everybody who handled that sample since they had it, anything that sample may have been stored with. But one of the big problems with the Ketchum study was it was, in a way, premature in that the sequencing technologies have actually, in the last 10 years, have outpaced the advances in computer technology. So, how, computers get faster, cheaper, more powerful every 18 months, Moore's Law. DNA sequencing technology has outstripped that. And when the Ketchum study was done, it was sent to a commercial lab, and they just got sort of the raw data back. But they didn't analyze it very well, and the problem with those kind of data is they're very short fragments of DNA, and it's very easy to misidentify a short fragment of DNA because life shares so much together. We're about 99% identical to a chimp. So if I just have 75 bases of DNA, it's almost certainly going to be 100% matched to a chimp. And depending on how you look at the database, you might just pick the first thing that pops out of the database, but it might turn out that there's actually dozens or hundreds or even thousands of things that it could be. And they sort of did a very superficial analysis. And so a lot of their sequences that they claimed were this or that, when they were reanalyzed by other people, they said, well, no, it's actually more likely to be this. So what's it more likely to be, bear or dog or Bigfoot, when it matches bear and dog? Bear and dog are perfectly good, reasonable alternatives.
Yeah, and I think that the perception on the DNA studies were that, they had a lot of Sasquatch DNA. And if you remember Jurassic Park, where they completed the missing sequences with frog DNA, that, a lot of people in the Sasquatch or Bigfoot community think that, when they put this study together, when they were missing components, they just took other pieces of DNA that they had assembled, to have a complete DNA strand. But that's not really the case. I mean, it could have been a bear, it could have been a moose, all these types of things. But, looking at, those studies, I guess, it kind of jumps out that they're trying to complete an incomplete picture. Can you talk about that a little bit?
To me, that's sort of one of the biggest problems, and is what I think holds back so-called Bigfooters, is, they say they want to be scientists, but, we rarely are out there trying to prove something. We're out there just trying to study something, and we can never really prove anything. We can just disprove as many of the alternatives as possible. kind of Sherlock Holmes-like, once you've eliminated all of the other possibilities, the unlikely becomes likely kind of thing. But if you're trying to actively prove something, it's really easy to have either conscious or subconscious blinders. You're going to see what you want to see if you're actively trying to do that, as opposed to let's collect the data and see what falls out.
So Todd, maybe just kind of shifting gears in terms of, your upcoming schedule. Are you working on any TV or media productions that the audience needs to be aware of?
Just did for Josh Gates on the Travel Channel, Expedition Unknown. He did a 4-episode thing on the search for the Yeti. So that was the last thing I actually did TV. I did a few forensic shows in between them and stuff like that. No, right now I'm actually concentrating on my real research. My postdoctoral research associate and I are about to start sequencing 40 complete monkey genomes. We're basically trying to fill in the missing parts of the primate evolutionary tree. So people have done a few non-human primate genomes for biomedical research and stuff like that. We're trying to fill in the tree so that evolutionary biologists have sort of raw data to continue on. So that's one big project. I'm also working on environmental DNA. So you may have heard of some of these studies where people go and scrub the subway seat or an ATM screen and find out how many millions of different bacteria and pathogens are out there in the environment. Think of an ATM screen. How many people a day touch that with their bare fingers?
Yeah, I think about it, just with my debit card.
Yeah. What are they leaving behind? And there's been studies done on that. So we're learning how to do those, because what we're excited about in my research group is something called environmental DNA. So in theory and in practice, we can go out to the forest and take just samples from the soil and sequence every single scrap of DNA and identify just about anything that has been in that area. Because again, we shed DNA constantly, and the new DNA technologies are so hypersensitive that if you go, say, to a pond or a stream and you take, a cup of water out of it and you extract every fragment of DNA in it, you can identify 99% of all the things that live in that pond or in that stream. Every species of fish and frog and snail and stuff like that. They have done this. They're testing this on soil samples. So we might be able to, instead of setting camera traps everywhere, we might be able to go just take a couple hundred samples of the soil in a forest and see what's living there. Because
that's really fascinating. Yeah.
So that kind of work is possible now, but it's getting the funding to do it is going to be tricky. If I'm going to a forest in Africa and I say I'm looking to see how many different species of nocturnal primates live there, I'm likely to get funding. If I go and say I'm going to go to Oregon and look to see if there's any primates living there, nobody's going to fund me.
So Dr. Disotell, assuming you had the funding for that, is there any kind of swag on kind of how long it would take to perfect that type of study or science?
It's probably good enough now. It gets better every 6 months. So, doing that next year or the year after is probably feasible. I suggested that to Spike, but they didn't want to deal with the cost of doing that. Because it is, that's the many thousands of, that's the thousands of dollars a sample, not the, hundreds.
So Dr. Disotell, do you and your colleagues ever think about your place in the universe? I mean, here you are on the forward reaches of science. I mean, to date, there's never been any scientists that have been this far forward in time. I mean, you have the best technology, you work at one of the best universities for this, but do you ever kind of ponder your place in the universe, and do you think the scientific community really understands that they have an obligation to move new discoveries forward?
I like to think that all scientists are more or less that. Again, some are more traditional and plodding, but I would like to think all of us are a little bit of that, because the one thing I like about science is, it's sort of self-correcting. When we do get things wrong, eventually we turn out, somebody shows us that we were wrong, and then we have to accept that. And if you're not willing to accept that I was wrong thing, then you shouldn't be in science. There are scientists like that, but they shouldn't be in science. I've been wrong, and, I've written articles that my newer articles absolutely contradict older articles, but I say that in the article. In 1997, we thought this based on this evidence, but new data has caused us to do new tests and new evaluations, and we were completely and utterly wrong in 1997. I've said that in print.
So Todd, just to date, kind of cutting to the chase, as the DNA judge, but have you seen anything to date that really has you stop and wonder, about the possibility of the existence of Sasquatch?
Secretly, I think it would be cool if it was out there. To be honest, to date, no. And I've been probably closer to the so-called evidence than most people, because, I've had people, lots of different people bringing me evidence. So it's not just what I can trump around the forest myself and find. I've had people, good people, bad people, mediocre people. I've evaluated lots of people's evidence, and I have not seen one thing to date yet.
And of course, the key word there is yet. So, we'll keep the faith, and certainly appreciate you, of course, continuing to sample and participate in research and discovery. And, who knows what we might discover along the way.
Years ago, I can't even remember which network, I did look at a Yeti sample from either Bhutan or Nepal. I'd have to go back to my notes. And we identified it as a Himalayan goral, a particular species of mountain goat. But it turned out where our sample was from was actually outside of the known range of that species. So the range of that species actually became bigger because a Yeti person had collected a sample of it.
And that's totally awesome. It's totally awesome.
So I guarantee you, and so is what's strange is there's no Bigfoot in Africa. No Bigfoot myths in Africa. There are other myths. They're all over Europe, Asia, North America, but Africa doesn't have. But they got plenty of other things there, walking around the woods. But if the level of interest and the number of samples of North American Bigfoot, if those had been collected in Africa, I guarantee you we would have found a new primate, because we've already found multiple new species of primates in Africa in the last 20 years. And so if people were looking for it in Africa, it would be beneficial in turning up new unexpected things. The fact that it hasn't yet in North America. The other thing in North America that I'm surprised it hasn't done, but again, not a lot of DNA analysis has been done. I've probably done, 90% of all the DNA analysis of so-called North American Bigfoot samples, and that's only, dozens to hundreds. But if somebody was really looking hard in North America, we might find new subspecies of deer or other things out there that we hadn't known, or people knew that, there were squirrels up on that mountain, but they didn't realize that they were a new subspecies or a new species of squirrel. We'll find stuff like that as more of these studies have been done. And like if we do environmental DNA studies, we might find that.
Well, Todd, we really appreciate you coming on Sasquatch Syndicate and concluding, part 3 of Building the Citizen Scientist. Of course, we're doing our best to educate our listeners on the best way to collect the evidence.
Make it clear to your listeners, I'm not looking for things in the mail.
And certainly it's, better to send them to the right place. And I think we got the name of that lab, Paleo DNA Labs. So
It's in Ontario, I think. The one at University of California, Davis, but I think they shut that down. Because some people just need to know, is this coyotes or dogs or wolves that are in my hen house, kind of thing. And so there is, use and need for it, or what subspecies of deer is actually on my land. So, that kind of stuff is done.
So again, for the researchers out there that may have evidence, that would like to submit evidence, you can contact Paleo DNA Laboratory at 866 DNA Labs. They're located in Thunder Bay, Ontario. So, Dr. Disotell, is there any final thoughts you'd like to share?
Well, again, educate yourselves, So, again, get an anatomy book. Read about how evolution works, how populations survive for a very long time. Actually do some scientific training or background reading. And it doesn't have to be deep and in depth, but know the difference between a deer bone and a dog bone and a bear bone when you're out there in the forest, so you don't all get all excited over a dead deer. And, again, read about evolution. And, if you really believe in Bigfoot, try to figure out where do you think in the evolutionary tree Bigfoot lives.
All right. Well, this has been a fantastic hour and a half. And Todd, really appreciate you coming on Sasquatch Syndicate. I know our listeners do as well. From the entire community, thanks again for testing our results, and keep up the good work.
Okay, take care.
This concludes our April 2017 podcast. Sasquatch Syndicate will return on Monday, May 1st. Thanks to all our listeners and those that have been out to our website and those following us on social media. Sasquatch is a controversial subject, so for all the believers and disbelievers and those that will tell you they have all the answers, just remember we're flying through space at 700 miles per hour. Buckle up.